we'll be going over cancer. There are two types of cancer, and also we'll be going over proto-oncogene and tumor suppressor gene. So let's start off with the basics of cancer. Looking at the picture here, that's your normal cell division. Your cell divides, and now over there you have uncontrolled cell division. It does not stop dividing. That's from GCSE. So we'll be going over what is tumor and what is a benign tumor and what is malignant tumor. So these are three definitions that we should know. Tumor. This is a 2-mark question. What is a tumor? The tumor is a mass of cell. So it's a mass of cell as the first thing. And then second mark will be going towards the fact saying that it's uncontrolled cell division. You won't get a full mark if you just say it's a mass of abnormal cell. Because abnormal cell can be different types of abnormal cell. What makes them tumor? What makes them bad? Is uncontrolled cell division. So it's a group of abnormal cells. These cells normally they look different. So this is a mass of abnormal cell with a feature uncontrolled cell division. That's the tumor. You can have benign tumor and malignant tumor. Benign tumor, exactly same definition here. It's a mass of cells with uncontrolled cell division. But what makes them benign? It does not metastasize to form other new tumors. It does not spread around the body to form secondary tumors. Malignant tumor does. So everything remains same in the definition, with the addition that it's a mass of cells, we can say, with uncontrolled cell division, but this time the mistake here, it doesn't. It does. There's a mistake here, there's an error there. So it does metastasize to form new tumors. So malignant tumors, it does move around, spread around to form secondary new tumors. This table compares benign tumor and malignant tumor, and you can have a read of them. This is from the AQA textbook, but I believe this is the main 3 points that we should be coming out with. Number one, benign tumor is different from malignant tumor. Why? Number one, benign tumor does not cause cancer. Number two, it does not invade other surrounding tissues. Number three, it does not metastasize around the body from secondary tumors. I'm showing you an image of a person with breast cancer. This is the PET scan, and we can see the black dots are the cancer, are the tumor, and they have spread around the whole body. So this is a malignant tumor. Tumors, so this is a slide which is showing you basically how the shapes of a normal cell and a cancer cell looks like. They are very different. Just for your own interest, you won't really be asked about this much or at all actually. This slide again showing you about benign tumor and malignant tumor, comparing them both. You should know the basic comparison, which we already went through. Now going over this, so this is a very important and new sort of topic in this video, and that is about how does cancer start, how does it happen, what causes uncontrolled cell division. To understand this abnormal uncontrolled cell division, we need to understand the normal cell division. How does it happen normally? So in here I'm showing you a normal cell, and this cell is called a fibroblast. Now you have these growth factors. These growth factors they bind to a receptor on the cell surface membrane, and when they bind to that cell surface membrane receptor, it initiates cell division. So in here I have 2 new fibroblast cells, so it promotes cell division. What really happens, the full story is this: you have proto-oncogenes. So these are genes. What they do, they code for growth factors. So it causes the cell to make lots of growth factors. And these growth factors, they go around the body and they're attached to receptors of cells. When they bind to the receptor of cells, it causes the cell to start dividing, to undergo mitosis. This is what happens normally. So proto-oncogenes, they promote cell division only when the cell needs it. That's healthy. When the cell needs to grow, when the cell needs to repair, just have proto-oncogenes making lots of growth factors. Growth factors bind to cell receptors and promotes the cell to undergo cell division. That's healthy and fine. What will happen if this happens constantly, non-stop? That's called uncontrolled cell division. That is bad. That's cancer. So we're going to see how that happens. Okay. So your proto-oncogene, we know that it's only, it codes for growth factors, right? It is only expressed, it only makes growth factors when the body needs it. Now, when it becomes, when that proto-oncogene does not work properly, meaning that it undergoes mutation and it is permanently on, is constantly making growth factors. In other words, there's constant cell replication. That proto-oncogene, we call it an oncogene because it has lost the role. It's doing too much. And the way I think of it is basically a car. You can think of the accelerator pedal as your proto-oncogene. The accelerator, you only press it down only when you need to go, and you let go of it when you want to sort of slow down or you want to stop the car, right? What would happen if the accelerator was switched on permanently? The car will keep going, and that is called an oncogene. So these are the text. We can make notes here. So I'm just showing you guys what is a proto-oncogene is. This is proto-oncogene. These are normal genes. They help the cell to grow, but when they mutate, keyword mutate, they become activated permanently, not temporarily, forever, permanently. So that mutated proto-oncogene has a new name, oncogene. So cell goes out of control and starts to divide and so on. So I'm showing you here proto-oncogene. When it has mutated, it becomes oncogene, and it leads to constant cell division. RAS is an example of a proto-oncogene that has mutated into oncogene. In the exam, they can change these oncogenes to test you, but the concept remains same. This is the exam question. Have a pause, read, and then try to answer the question. So I'm going to read the KRAS gene. Now, first time you've seen this, doesn't matter. KRAS gene, it's a gene. Okay, what does it do? It makes a protein, or it codes for a protein, KRAS. So this is the KRAS protein. This is the phenotype. This is the genotype. Okay, that protein relays signals from outside a cell into a cell's nucleus. So it's somehow giving signals to a cell's nucleus. And what that does, it stimulates cell division. Keyword, it is stimulating cell division. So KRAS is linked to cell division. Now, an alteration of the KRAS gene produces oncogene. So now we know what this is. KRAS is a proto-oncogene, and when it becomes alteration, means what? Mutation. When it changes, it becomes an oncogene, and this causes a tumor to develop. Now, this is part of our spec. We are linking proto-oncogene to oncogene, although KRAS is a something that we haven't heard of, but doesn't matter. We can link it up. Suggest, give a reason, scientific reason, explain scientifically how an alteration. So the keyword here, alteration. Okay, they want to sort of confuse you. What's alteration? It means change. What does that mean in science, in biology? Mutation. If they say mutation, it might be too easy. People might think, oh, it's talking about proto-oncogene to oncogene mutation. So they use alteration. So mutation, okay, in the gene can cause tumor to develop. Have a pause. What would you write? Okay, so 3-mark question. Okay, so you're going to say number 1, mutation. Remember, you have to mutation in the it happens. So mutation happens in the KRAS gene. Okay, and this basically will cause what? Obviously the normal function of the KRAS protein. So KRAS gene is mutated into. So the KRAS protein is now different. What's the normal job of KRAS protein here? It says it gives signals to stimulate cell division. So this is a proto-oncogene to oncogene. So it's going to continue the same job, but at more constantly. So it's going to continue signaling, but what? More signaling. It's going to do more signaling. So change in the signaling protein. So the protein will change and will cause more signaling. In other words, what will it do? It's going to stimulate more cell division. So that leads to rapid uncontrolled cell division. And this is why you form a tumor, uncontrolled cell division. We're going to move on to the next thing, tumor suppressor gene. Now, tumor suppressor gene, you can think of like the brake of a car, brake pedal of a car. So you stop the car when it's when you want the car to stop, right? So in the same way, our cells have tumor suppressor gene. Thank God we have tumor suppressor gene. What they do, they are the caretaker of the cell. They are the gatekeeper of the cell. So let me explain how. These things you don't need to know really, but just sort of giving an idea. So let's say there's a damage in the DNA. The DNA is damaged. Okay, that cell, this tumor suppressor gene will basically try to repair it. Okay, will fix the problems, and then if it's too bad, if the problem is too much, it's going to cause the cell to die. So in other words, it takes care of the cell, it repairs the cell. If the damage is too much and is dangerous, it's going to cause cell program death. Cell will die. So this is a very important thing of cell. We don't want bad cells to start replicating and replicate and become more. We want it to finish off. If it can't be repaired, kill it off. Sort of apoptosis, cell, kill the cell, destroy the cell. So therefore, it's better, because you don't want that cell to divide more and more, and after some time you have thousands of bad cells. So tumor suppressor gene. So these are in cancer. What happens to these cells? So remember, we said these are the brakes. So in cancer, what happened? These genes are not working properly. They are switched off. So now we have a car, accelerator is on permanently, that's your oncogene, and the brake is not working, that's the tumor suppressor gene. So the tumor suppressor gene is not working, okay, it's inactivated. And we mentioned already what it does, okay, they slow down cell division, they repair the DNA mistake, and then if it's too much, it tells the cell to die. Now, there are many examples. So tumor suppressor gene is a generalized word. So these are the specific words. TSG stands for tumor suppressor gene. Examples are P53. So P53 is found in almost 50% of cancer. So this is the tumor suppressor gene that is not working, that is inactivated. Breast cancer have BRCA1 and BRCA2. These are the two tumor suppressor gene that is mutated, that is not working in breast cancer gene. So tumor suppressor gene, why are they not working? Because there's a mutation, and that mutation caused that tumor suppressor gene to be inactive and not working, turned off. So these are BRCA1, BRCA2. These are mutated versions of tumor suppressor gene. If mainly a female has these genes which are mutated, they have high risk of getting breast cancer. And P53 is also a tumor suppressor gene. If that gene is mutated in your body, you have a risk of having a cancer. Focusing on estrogen and breast cancer, we've spoken about previously about estrogen is linked to breast cancer. We know that estrogen is a hormone, it's a steroid hormone. It enters the cell, is lipid soluble, but it diffuses in, and then what it does, it binds to the transcription factor, and then it initiates cell division. It promotes transcription, and also it promotes cell division. So therefore, that's why estrogen is linked to breast cancer. Now, breast cancer is the most common cancer in the UK. These are a few facts which you don't need to know for exam, but it's very, very important, I guess, for common knowledge. Family history is very important for breast cancer, and also having these two genes in your body is also a risk factor as well. This is an exam question about tumor suppressor gene. Pause the video, find the keywords and have a go. Okay, so this is how I would read it. So this is a syndrome, okay, Lee-Fraumeni syndrome. is caused by a mutation. This syndrome is, so it's a name that I haven't heard of. It's fine. So that doesn't really bother me. It could be any syndrome, but the science is still the same. It's caused by mutation. Okay, so this is by mutation affecting tumor suppressor gene called TP53. So okay, so the key thing here I have TP53, and what that is a tumor suppressor gene. And what is it? It's mutated. That means it's off. That means it's not working. So it's switched off. This gene codes for a protein that initiates cell division, cell death. So it's telling me the normal function. Normally this gene will cause cell death when the DNA is damaged and can't be repaired. Now, if it's mutated, it leads to the production of non-functional protein. Suggest how the non-functional protein may lead to cancer. In other words, suggest how this TP53, which is inactivated, not working, can lead to cancer. So you can say, number one, there's a mutation, okay, which changes the DNA of cell. So you have a mutation, and that will change the DNA of cell. Now, the damaged DNA is not repaired. Okay, so you have a mutation happening in the cell. Okay, normal cell. And normally, what would you have? If you have a mutation in your cell, that DNA would be damaged. And what will you have? You have the TP53 fixing that. If you can't fix it, it's going to cause cell death. But the problem that we have here is that it will not do that because it's switched off. So it leads to cancer. How? You have a cell, and in that cell, obviously, you have tumor suppressor gene switched off. So when you do have a mutation happening in the DNA, the damaged DNA is not repaired and the cell lives, is not killed. This mutation can lead to uncontrolled cell division. So you may get a mutation in a particular gene which causes uncontrolled cell division, which promotes cell division. So therefore, this leads to cancer, okay? And then some of these tumors, these mass of cell, can spread to other parts of the body, causing cancer.